Hi, this is Sakurai Carolus, Science, and we're here today to learn about
Cellular
respiration.
Hi guys, William Kettle here, and today we'll be talking about cellular respiration. Basically, cellular respiration is how your body makes energy. Intaking glucose and oxygen. Glucose is C6H12O6, and oxygen, which is 6 oxygens, so 6 O2s. The outputs are carbon dioxide and water and ATP. Today we'll be talking about cellular respiration, glycolysis, pyruvate oxidation, and the citric acid cycle, and the electron transport chain. And that'll be the basic overview for today. So let's get into it. The first step of cellular respiration is glycolysis, and is the simplest part of cellular respiration to understand. So it starts with 1 glucose and uses 2 ATPs. Now the 1 glucose is, like I said before, C6H12O6, and it breaks down that carbon chain of 6 into 3 carbons. And now 2 of the same reactions happen to create 1 pyruvate. So I'll be describing one of the reactions, but just keep in mind this happens twice to end up with output 2 pyruvate. Now what happens is the 2 ATPs separate the 6 carbons from the glucose into 2 groups of 3 carbons. And then the 4 of the hydrogens from the H12 are broken down into 2 groups of 2 hydrogen and then combined with NADH, NAD plus molecules to form 2 sets of NADH molecules, each with a hydrogen. So the product of this reaction ends up being 2 pyruvates and 2 H2Os. So now we move on to the next concept, acetyl-CoA synthesis and the Krebs cycle. Now, acetyl-CoA synthesis is a continuation of the previous process, glycolysis. Acetyl-CoA synthesis, also known as pyruvate oxidation, uses 2 pyruvate molecules that were products from glycolysis to form acetyl-CoA. Remember that a pyruvate molecule has 3 carbons. What happens is that first, a pyruvate loses 1 carbon molecule when it oxidizes in the form of CO2. The leftover 2 carbon molecules help make up an acetyl group. This acetyl group binds with an enzyme called
coenzyme A,
or CoA. During this process, an NAD plus ion reduces to form an NADH molecule to be used in the electron transport chain, which I'll talk about later. In summary, acetyl-CoA synthesis is when each pyruvate molecule from glycolysis oxidizes and produces an acetyl-CoA molecule, a CO2 molecule. You should know this kind of stuff. And 2 NADH molecules. Moving on, acetyl-CoA synthesis starts with a bigger cycle called the Krebs cycle, or the citric acid cycle. In this cycle, there are 8 main steps that work towards using this newly generated acetyl-CoA molecule to help produce a lot of ATP later on. Remember, that's what the whole goal is here, energy. To start off, this acetyl-CoA molecule will merge with
oxaloacetic acid,
which has 4 carbon molecules. Remember that acetyl-CoA has 2 carbon molecules. This is important stuff, guys. Take notes. These will combine to form citric acid, which has 4 carbon molecules, plus 2 carbon molecules suggests would have 6 carbon molecules. After this, the citrate becomes isocitrate. It's isomer. With the help of the enzyme
aconitase.
No, it still has 6 carbon molecules. Then the isotrate oxidizes to become alpha ketoglutarate. In this step, CO2 is released, making the alpha ketoglutarate have 1 less carbon molecule, so it would have 5. Also, the NAD plus reduced to NADH in this step. Next, the alpha ketoglutarate becomes succinyl, CoA, where once more an NAD plus molecule is reduced to NADH and CO2 is released, making this molecule only have 4 carbons. Later on, this acetyl-CoA becomes succinate, generating a molecule of GDP or ATP, both functioning as energy sources. Then, succinate becomes fumarate, producing a molecule of QH2, which is a molecule used in the production of FADH2. Afterwards, the addition of water, the fumarate becomes malate, still a 4-carbon molecule. Lastly, the malate becomes
oxaloacetate.
Remember, this molecule from the beginning of the cycle should also producing NAD plus to NADH. To get an overview of what has occurred in the cycle, remember that we are zooming in on an activity of the acetyl-CoA molecule, which is formed from one of the two pyruvate molecules produced during glycolysis. With this acetyl-CoA molecule, 3 NADH molecules have been made through reduction of NAD plus ions. 2 CO2 molecules have been released, and 1 FADH2 and 1 GDP molecule was produced as well. Remember, this is important, guys. Remember that in glycolysis there are 2 pyruvate molecules, which means that there would be double the amount of products for 1 glucose molecule. In the next section of this video, you will see how these generated molecules are crucial in forming the abundance of ATP in one's body. The next part of our process is the electron transport chain. The key concepts of this is that this is a process that creates most of the ATP in cellular respiration, the thing that we really need. The Krebs cycle creates 10 NADH molecules and 2 FADH2 molecules. NADH creates a little more ATP than FADH.
2.
NADH, FADH
2.
goes through oxidation that releases high energy electrons and protons. This is important. The high energy electrons then goes through coenzyme Q and cytochrome C to lower the energy level because they're going from a high to low state. The electrons will go through electron transport chains starting with complex 1
Which NADH electrons go through?
or complex 2.
Which FADH2 electrons go through? NADH electrons form roughly 3 ATP. FADH2 electrons form roughly 2 ATP. This is due to the different complexes that they've released their electrons into.
As the electrons move through complex 1 through complex 4, the hydrogen ions are being pumped out
Into
the intermembrane. Then the hydrogen ions will come back into the matrix inside the mitochondria through a protein called ATP synthase, using energy from the previous release of energy from the coenzyme Q and cytochrome C. The equation can be seen here, as observed. The hydrogen ion that goes through the ATP synthase will allow ADP plus phosphate group to create ATP. This will create roughly 32 to 34 ATPs, which is what we want. In the last lab we conducted, we observed peas, and one trend was that the peas that were germinating respired the most, meaning that peas that were attempting to grow out of the ground, as dark surroundings indicated, require a large amount of energy that is produced from cellular respiration. Since the peas had a storage of organic molecules to help them grow, since there was no sunlight that would allow for photosynthesis to occur, they could use stored glucose molecules to carry out the process. With the production of ATP, the pea would still be able to use this via the breaking of bonds to grow. Last, we have to have an overview of cellular respiration. So, here's some key points. The glucose obtained from photosynthesis or their consumption of another organism is broken down through glycolysis, which is able to happen because of the presence of oxygen, O2, from the environment. The glucose is broken down into 2 molecules of pyruvate outside the mitochondria. Pyruvate enters mitochondria with the presence of O2, where it then undergoes pyruvate oxidation, where a carbon is broken off the pyruvate. The carbon that was broken off oxidized, producing CO2 as waste, which is one of the products of cellular respiration. The rest of the pyruvate bonded with coenzyme A, forming acetyl-CoA, which is then broken down more in the Krebs cycle, producing CO2 as waste products, also producing 3-NADH and FADH2. The NADH and FADH2 produced from the glycolysis in the Krebs cycle are then oxidized, releasing electrons through the electron transport chain as a series of proteins. As the electrons go through the protein pump, out H plus ions, which then go through another protein called ATP synthase, where ADP is bonded with the phosphate group, creating ATP, and where H plus ions are oxidated, forming H2O, the other product of cellular respiration. Well, thank you guys a lot. It was a blast. I hope you know more about cellular respiration. Have a great day.